That's my Mia Maria. Okay. Okay, guys, I'm gonna do a quick video here on your practical 2. You know, you had a mock practical for practical 1, so this one's gonna be a little bit different. I thought I'd give you some differences about it and that kind of thing. So, right, that's my other class. This is the biology lab website. In the content section, right, there's the PowerPoint for practical 2. I think I've been using it. That's what you should be using to study for practical 2, right? Hopefully you use practical 1 PowerPoint for practical 1. And so if you look in the grade book, there is, if you notice, practical 2 is broken down into 2 items, right? Yeah, practical 2, 80 points, which you'll earn for the first 70 questions. And then after you're done with those questions, you'll complete, hopefully correctly, 2 genetic problems. So I just want to make it clear what that will entail for this upcoming exam. Okay, so the first 70 questions will be just like the first practical, where you go from station to station and answer questions based on, you know, say looking at figures. So you might see something like this for a figure, and it might ask you, well, what phase is this arrow pointing to, or what phase is that pointing to? You'd have to remember these are metaphase, these are anaphase, telophase, that kind of thing. You might go to a station and you'll see this picture, and the first question you get, well, what phase of mitosis does this model represent, right? It's telophase. And then there might be arrows pointing to some of the things that are pointed here, and then you'd be expected to know, you know, what they are, spindle fibers or cleavage furrow. This is, sometimes people mistake this picture with this picture. They call this one on this side telophase when it's interphase. But at this point, at interphase, you now have 2 complete cells, right? You're done, right? And then chromatin refers to the DNA during interphase, where chromosomes refer to the DNA during mitosis, right? So prophase, metaphase, anaphase, telophase, all of it. There's arrows pointing to any of those, like they are here, this chromosome, right? Then that chromosome, it's not chromatin, then you call it chromosome. Okay, so those little things you have to, you know, keep in mind when you're doing this exam. You know, if you display the chromosomes here in metaphase, what do you refer to that as? Right, that's what a karyotype is. It's a display of all the chromosomes from a cell that was in metaphase, right? You're able to arrest the cells at that point, and then take a picture of them. So know the difference between the cell cycle, right? You have interphase and the mitotic phase. Those are 2 phases of the cell cycle. And then mitosis has 4 subphases. Okay. So when you get done, like say, getting questions about this picture, or what, right? This blue cap thing is called a volume meter, right? You get questions about that, right? It measured oxygen consumption. And then I might ask you to identify these pigments here that you see, right? The top one's carotene, xanthophyll. First one's chlorophyll A, then chlorophyll B. They looked a little bit different when we did them in the lab, but this is a pretty good, you know, representation of it. So I'll probably use that. Now there are 2 corn models, right? These are corn on the cob, and they're helping us to learn about meiosis. So like the top 2 here are P1, these 2 are F1, and the bottom would be what the corn would look like under the F2. This is showing phenotypes. So they call it on here at the bottom, you see purple aileron and colorless aileron. You could call this purple or yellow as well. But when you take the practical, you might see this, and it'll help you answer the question. So either way, if purple and yellow you're easy to remember, that's great. But you'll get some questions about that. And you'll also see this on the practical. So there'll be 3 questions about this particular picture or these corn right now. This is a dihybrid where we're looking at 2 phenotypes or 2 genes instead of the monohybrid, which we only looked at 1. So question 70 will be worth 10 points on the practical. And it'll ask you what are the phenotypic ratios. Okay, so that means you need to use phenotype, not genotypes. Right? They're written in genotypes at the bottom here. But there's 9 in the F2. There are 9 that are purple and round. There are 3 that are purple and wrinkled. And there are 3 that are round and yellow. And there's 1 out of 16 that show both recessive phenotypes, which would be yellow and wrinkled. So it's a 9, 3, 3, 1. I'm going to talk about this in the lab, before the practical, and so hopefully if you got any questions, ask about it then. I'll emphasize that what they use here for genotypes, they use big R, would be no different than, you could use big A for that. If they use SU, it's capitalized, right? That would be the same thing as, say, using 2 capital B, BB, right? It's the same as capital SU. If they're lowercase r and lowercase s over here, then it's the recessive wrinkle phenotype, right? Capital letter S, little u is round. Capital R, little r, colored or colorless. They have colored aileron and colorless aileron. But you could use these letters to denote the same way, as long as the upper and lower cases. That's what. But anyway, I just wanted you to understand. So what you'll do is you'll answer the first 70 questions, which will be worth 80 points because the last question is worth 10. Once you're done that, you turn that in, and then you'll be given 2 genetic problems. Now, I'm going to make it easy by telling you that the 2 genetic problems that you might see is either going to be question 1 on the Genetics 3 lab. This is the Genetics 3 handout lab. I actually combined both Genetics 2 and 3 together. So if you look into the content section, right, you'll see it as just one, under lab handouts. There's there Genetics 1 handout, and then 2 and 3 are together. But under the 3, right, you have this is Genetics 2, and then it turns into Genetics 3. Under this, there are several questions. I think it's the first 8 questions, or 9 questions rather. Right, you could get this question too. The first 9 questions, or is it 10? What is the genotype? What is the type for the F1? The first 10 questions. And the question will tell you need to write down a cross, which would mean, in this case, a woman who's heterozygous means, right, if you're using brown eyes, that means she'd be big B, little b. Blue eye would mean 2 little b's. So you get 2 points for the cross. It'll say it on the when you take the exam, right? And you get 4 points for the Punnett square and 4 points for the genotype. So you get 2 of these questions from question 1 to question 10. Those will be selected. Different lab sections may get different questions. So So as long as you know how to do these 10 genetic problems, then you should have any trouble getting both of those right on the practical. Okay, so just wanted you to give you a heads up on that in case you don't have me for lab. Mostly is why I'm doing this video. So good luck with it.